Hello, I'm here with Don Pfister, and we're at the Mycological Society of America meeting on the campus of the University of Florida at Gainesville. And we're in the Hilton Hotel at the edge of the campus. And we are part of the project for the oral history of mycology. And Don and I have been doing this for several days, and today we're here to interview Hannah Reynolds. And so, Don, you have a question.
Great, thank you, Meredith. And we're here with Hannah, and we kind of have you introduce yourself and then talk about your early background, your training, how you got to fungi.
Your family,
general...
were they interested in things like this?
Okay, so like really early.
Yeah.
Okay.
Right now with where you were born.
Okay, so yeah, I'm Hannah Reynolds, and I got my start in science, I guess, just enjoying nature. My family weren't scientists, but they did value books and nature a lot. And so we'd watch nature programs together, we'd go on little hikes, we had lots of nonfiction and fiction books. And they always encouraged me to do, whatever I was interested in. But it wasn't until late in high school that I thought I might be a biology major. So before then, I was a little bit more excited about like writing and art. And but I got really excited about my biology class in college, and also in high school I did a couple of summer experiences. So one was actually back in middle school. I did a summer camp where we got to do embryology, and that was the first time I had dealt with really nice lab equipment, and I really liked that class. And
Where was it?
That was in Denver.
Okay.
They did a little experience for middle schoolers. And then in New Mexico Tech, which is in Socorro, New Mexico, which was about an hour north of where I lived, in sophomore summer of high school, I did an engineering camp. And this was a little week-long experience for environmental engineering. And I found my favorite thing in the class was microbiology. So we learned the nitrogen cycle and and the sulfur cycle and how that could degrade a sewer system. But I thought from that experience, I don't really want to design a sewer system or a landfill, but I'm really interested in these microbes, and maybe I could be a biology major. So then in college, I started my biology major. I really liked it. And the first year I was looking for jobs, and I heard from some professors that you could get jobs working for these different labs, because I would look for ways to make money. And I applied for one job and didn't get it, but then I applied for my second time. I applied for a lab assistant job. I got it, and this was working for Cameron Curry, helping maintain the leaf cutting ants that he worked on. And at the time, my department had a lot of entomologists. I got excited about entomology. That was one of the reasons I was applying for his.
Where was Cameron at that?
That was in the University of Kansas, in Lawrence, Kansas. And I was a sophomore. And so because these are fungus-growing ants, I learned about fungi. And I took his mycology class. And I really loved these ants. I loved this symbiosis. So when I was getting ready to graduate, he and some other entomologists there encouraged me to look at different programs. And I looked at entomology and mycology programs. I did my interviews. I actually interviewed as well with Meredith at Louisiana State. And I interviewed at, I think, a total of 5 different universities, like 3 entomology programs, 2 mycology programs. And I ended up accepting a PhD-ship with Rita Svilgales at Duke. And at the same time that I was looking at all these programs, I was also applying for a Fulbright to use my French minor, because I wanted to go live in France for a year and do science there. So I got that, and so I delayed starting graduate school so I could go to France and do research on the fungus-growing ants that an entomologist was studying there. And then after that, I started graduate school at Duke.
Somebody in France was studying atta ants or
Attine ants, yes. They had Acromyrmex and atta, and that was kind of leftover colonies from a graduate student's project. They were a social insect behavior institute within a science college.
So how had they got them there? Was there a lot of problem with permitting?
I don't know. They had been in the lab at least 2 or 3 years by the time I got to work with them.
And they came from
They came
Panama,
originally, I think,
or-
from Panama.
Mhm.
Yes. So I don't know how they ended up getting the permits, but I know after I did my project there, they ended up moving the colonies to their local museum and tours.
Oh, okay.
And the thing about the ants is that the colony just grows and grows and grows until it's unmanageable, and so you have to either keep culling it and feeding it, and or you have to put it someplace where it can really be maintained all the time. And so that's the museum volunteered to.
It'd be a great museum.
A beautiful resource, yeah, and especially for some tropical insects in chilly northern France.
Yeah.
Yeah, and it would be great too to show one of these movies. I remember as a kid seeing a movie where army ants went through the place and wiped out everything.
Yeah, they're fascinating animals and amazing behaviors. So I had a lot of fun at that project. Then I started with Reynolds, and I ended up moving a little bit more away from entomology into just classic mycology, and I did a systematics and phylogeography project on Elaphomyces truffles for my PhD work. And then while I was in the middle of that, I got invited by a friend to go on a caving trip. He liked caves, and I had a car, and this was an undergrad who didn't have a car. And so he wanted to drive to Virginia to go on a caving trip, and I did, and because I was curious. And while I was on the trip, it was several hours underground with some caving experts who showed us around a set of caves. And they told me about, when they heard I did mycology, they got excited because this disease of bats had just kind of broken out a couple years ago. And they wanted me to study it, but I said, I'm in the middle of my dissertation, but maybe someday, And so when I started getting ready to graduate, I looked for jobs studying white nose syndrome. And I was lucky enough to find one at the University of Akron, where Hazel Barton, who's a cave microbiologist, had got a grant to study white nose syndrome. And I wanted to study it from the fungal angle because at that time, we really didn't know much about it. There had been a couple of 2 or 3 really good fungal papers, but I wanted to look at some of the ecology of it. So she and I got to look at some ecological and physiological traits of this fungus. And then after that, I can't remember if it was 2.5 or 3 years, I moved to Ohio State to work with Jason Slott and we were looking at comparative genomics. So I learned, my progress, I was like, kind of started doing ecology and, ant behavior and a little bit of fungal work as well with Cameron, and then did more ecology and behavior, and then I learned in Reynolds' lab all about the molecular side, like my first DNA extraction, my first PCR, sequencing, phylogenetics, all those tools. And then with Hazel, I did a lot more classic microbiology, like careful microbiology experiments. And then with Jason, I got to learn genomics, so genome analysis tools. So it was kind of my path. And then, then I was looking for faculty jobs, and actually, how much information do you want about my job search?
Oh, I think it's good for folks to hear what it's like, what it was like.
True life story.
I was kind of toward the end of my postdoc with Jason, I just gotten married and I was pregnant. And so I thought, well, like my top priority kind of switched from let me do the most exciting science I can possibly do to let me raise my family and be stable, because I loved all my jobs and my experiences, but I wanted to be stable. And so I said, okay, I'm going to get a job one way or the other. I have a lot of skills to offer, but my main goal is now just to be able to settle down. So I applied for jobs, whatever jobs I could, industry, academia, and government. And so I just had a list of all the deadlines. I'd send applications out, and I got interviews at Sherwin-Williams headquarters because they had a microbiology program, and I got an interview at Western Connecticut State. And when I did these interviews, I was 8 months pregnant.
Did they know ahead?
Yeah, I did. I did tell them partly to get permission to go on a plane. But I ended up getting the job at Western Connecticut State. And then I moved there shortly after having my baby and started my job in December of 2017, and I've been there ever since. So my job there is to teach microbiology. It was a microbiology position. And when I was applying for academic jobs, I'd apply for mycology, microbiology, or genetics, because I felt like those are the big classes I could teach. Yeah, because undergrad-focused institutions tend to advertise for large classes, because those are the most pressing needs. And I thought genetics or microbiology would be something I would enjoy and be good at teaching.
Now, when you did the microbiology work, did that include bacteria too?
Yes. Well, when I did my research, I didn't work with the bacteria, but there were a lot of students who were working with bacteria in that lab, and I would sometimes advise them on just basics of media prep or extraction techniques, cloning techniques, things like that. So I felt like I was learning a lot of bacteriology as well, even though I was, my project was all with fungi.
Yeah.
So what's it like for you teaching at the Western Connecticut? What's the aura?
Well, it's a very a positive place. So it is a place where we are encouraged to be innovative and we really support each other. So we, in terms of the numbers of students, it varies because in the fall I teach the nursing department and they have all of their nursing second-year students take microbiology from me. So that can be up to 80 students. And then all the other classes I teach are more small, upper-level biology classes. And those tend to be around 20 students, sometimes as low as 12 students when it's our upper-level research class. So it's the size of the students. And I also, something that's kind of special about my university, which is a regional state university, and many other ones, is that the professors teach a lot of the lab sections. Since we don't have PhD students, we're the ones teaching the students how to focus the microscope, how to transfer bacteria, and so the students get a lot of hands-on attention from us because we're the ones really showing them how to do these things. So at the same time, so what the students see is, I'm there explaining things to them, helping them. But in the background, our faculty have some resources for improving our teaching. So I got to go to a summer institute for scientific teaching that was funded by my university. My attendance was, and that was a week-long program all about how to include active learning in the classroom, and we just encourage each other to try some new techniques. Sometimes they flop, but because we're not afraid of trying out new things, we can innovate in our teaching, and that's very rewarding for me.
During COVID, were you Zoom lecturing, or were you?
I was. We were actually, in March of 2020, our campus shut down, and then so I switched to not Zoom, but WebEx, which was our tool, right? And same general idea. And then I'd actually already signed up to teach a summer class. And I didn't know if anybody would want to take my now online-only summer class in micro, but it filled. And so I taught an online-only class. And that was fun because in the spring, did a lecture, and then I innovated with the labs to do data analysis. All right, so we did just data analysis, and they had already had the first semester of lab already. And then in the summer, I did some at-home labs where they would do fermentation projects. I had them make pH paper out of cabbage and paper towels. I had we had to try to get them to make their own aluminum foil petri dishes out of Jell-O. That was a bit of a disaster, but I pushed them and they pushed themselves and they did a lot of good work. And then in the fall, we had the option to return, but I decided to not because it was very up in the air when we would get to come back. And in fact, the campus thought we could start in August, then on-campus activities were delayed 3 or 4 weeks. So I was very glad I had decided to teach online that semester. And then in the spring, 2021, I went back all full-time and have been full-time online since.
It was such an interesting time. I taught 1 semester online, a general, my biology of fungi course, and I thought, how am I going to do a lab and get the appreciation here of what it's like to do this. And so what we did was to put together a little at-home kit, and we gave them a lot of stuff, and we gave them each a mushroom growing kit.
Nice.
And we had them do a lot of stuff, go out and look and bring things in and do a moist chamber. And
Nice.
what else? We had them make ginger beer, and we had special lectures on sourdough. So, you know, I just called it Mycology at home, you know.
Yeah. I did that, some of that, and in the fall, I hadn't had permission to send home petri dishes in the summer.
I know, they
but in the
didn't want
fall,
us to send a lot of stuff.
yeah, I got permission to do. I sent home petri dishes, and then they did tea streaks of yogurt, and then I also sent home little bleach tubes so they could sterilize their stuff afterward. That worked pretty well for at least just teaching the basic technique.
Yeah.
But it was, I don't know, see, you know, what was funny is students actually, they really rose to the challenge, and I was happy to see that.
Yeah, I think that was the case, that in many cases that they did. They found that it was fun. I found with Zoom, for example, that I probably got to know the students better in a certain way than in person. And even though I had a teaching fellow, I always did all the labs too, when it was live. But because you've got the names, and because you've got the faces in front of you, you can manipulate it a little bit better than you can in the classroom. So it's interesting.
My first 6 years teaching was at Hope College. And so there, I think I had like 37 hours teaching a week, contact hours. And I also had no TAs, but it was fun. We had they emphasized a lot of research projects. Do you have that kind of thing?
Yes, what we do, we have 3 general ways students can help with research in the normal academic year. So first we have student independent studies where the semester prior they have to make a little proposal, very short, of just what they're going to do and how many contact hours they'll have for it. And then when it's approved, they get to do it the next semester. So I've had usually 2 to 3 students working with me any given semester on a small project. And then all of our seniors have to do research. And the 2 ways they can do that is advanced senior research, where they've usually done a couple semesters already, and then they plan their project in advance and they do a senior thesis. And the other way is group senior research, where they just sign up for a class. They could have done no research up to that point, but they're a senior and they fulfill their other requirements, except for a few classes, and then they do research within a 12-person class. So I've helped students with all of those. So I've had the independent students and a couple of student theses, and then I've taught the research class a couple times.
Great.
They come up with their own project.
Yeah, so for me, I'm able, because I'm doing microbiology or mycology, I'm able to give them a lot of leeway. So I give them the theme, and then they come up with specific questions to test. So I give them kind of a big question, a few parameters, a couple papers, and then they come up with kind of the smaller, more focused questions and hypotheses. For my colleagues who work on vertebrates like mice, because they have to go through the IACUC approval, the professor has to do a lot more of the planning of what is the mouse going to experience. Yeah. And then he has a plan in advance that's approved, and then the students within that get to decide kind of how they're going to use the data that come out of that project. So he does mouse obesity studies, so he'll say, we're going to feed the mice this is how many mice we're going to have, et cetera. But then the students get to decide, like, what statistical tools will they use.
I think fungi are easier.
Yeah, the students have really liked the independence working with the microbiology, and a lot of them already have pretty good bench skills by the time they take my class, because they have to do genetics lab before they take it for that group senior class, and many of them have taken microbiology, so they have a lot of comfort with just pipetting and that kind of research. So we really get into it, and I'll like do a couple of field excursions. And then we'll do a lot of lab work, and then we do data analysis and explanation, literature exploration.
So you probably don't have any time for your own personal research.
I do a lot of my own research in the summers, and then we are eligible to apply for faculty release time. So I've been able to get some faculty release time, maybe 3 credit hours a semester. Often I'm using that to teach myself new analysis programs. So I'll say, if there's some software that would take me, several hours to install and use and learn that I think would advance my research with students, that's what I'll tend to use that for. And then coming back from COVID, I found that it was harder to recruit students for in-person work. So one student, she loved working with me, but she was doing an on-campus job, and the amount of swabbing and testing she was doing was kind of nerve-wracking for her. So we did an online project together. So I used some of my release time to do just keeping my culture collection going, things like that. Things that usually I have the student researchers do. Yeah.
Well, it sounds like a very lively and active program.
Yes, it is. It is, and we get to. I have enjoyed getting to chart my own course with that work because we have, it's just a positive place. So we get to try things, and sometimes they fail, but we're just encouraged to keep trying.
Mm-hmm.
Sounds good.
I think you've given people a very good idea of a trajectory.
I think so, and I think there are many people who've had a similar trajectory, right? There's a lot of jobs at small colleges or small universities, maybe that are focused on the undergrads a lot more, but still that research, even if it's a small amount of hours, compared to what we do in graduate school or postdocs, it still kind of keeps us fresh, gives us ideas, lets us share things with the students and with the community.
Yeah, great, good.
Sounds great.